I'm going to talk to you about the preliminary results of one of the studies I am involved, and it concerns, we're trying to develop a new assay to monitor transplants, not only kidney transplants though, heart, lung, and kidney transplants in Great Ormond Street Hospital. So a little bit of background. As you all know, solid organ transplantation, is a successful treatment for end-stage organ disease, but with a mean survival which is limited. So depending on the organ, you can see the numbers on my slide. And our main issue is that even though direct post-operative management has massively improved, then after the first year, the follow-up has not really changed over the last 30 years. The main problem is what we call chronic damage. It's the same for all the three organs, obviously. The etiology is poorly defined, but we suspect that it has to do with persistent mild asymptomatic rejection, which is undetectable. So what are we doing? The gold standard is a tissue biopsy. But this has some issues, though, as we all know. We have the site selection bias, that this might result in a false reassuring biopsy. It is something invasive, unpleasant. Our little patient, we have to put them under general anesthesia, which is not always easy, and is infrequent. We are not going to do everyday biopsy to patients. So obviously, we failed to detect important changes, minor changes, in between the biopsy points. Our current method is to use immunosuppressive treatment. We assume that is enough, but this has the failure of recognizing the natural variation of the levels between the same patient and between patients, failure to take into account compliance issues, as we have discussed already yesterday, and is a one-size-fits approach, which obviously is not optimal. So I hope that you all agree with me that we need new methods in biomarkers. So what the MODE study proposes, the primary aim is to develop an assay that, and establish the proof of evidence that we can use the donor-derived cell-free DNA, we can distinguish it from the patient, and we can monitor this with a non-invasive way, just by a blood test, let's say, so as to anticipate rejection before having to do a biopsy. The secondary aim, which will be for the next year's symposium, to present you the results, is to determine the potential for a larger UK-based research project, correlating that with rejection episodes, DSA antibodies, clinical outcomes, and immunosuppression levels. So the methods here, please, I want all your attention. I have to make you more familiar, for the ones who are not, with two main things. The one is the cell-free DNA, so it's stored DNA fragments that we can find them in various body fluids, from the urine, saliva, peritoneal fluid, and this is released from cells when they are dead, really, so after apoptosis, necrosis, or netosis. This was first described long time ago, but it had the first clinical implication, really, by our oncology colleagues on 1977, that they say, hey, guys, we might be able to use that for tumor diagnosis. And it was 20 years later, or 1997, that we finally applied that for prenatal diagnosis, and we're still doing that nowadays. The second idea that I want you to pay attention and recall your genetics is the SNPs. So it's something very modern nowadays. It is the single nucleotide polymorphisms. So it's basically a variation on our DNA sequence. And as you can see, instead of having an AT in your DNA, you will have an A nothing, a GC, an AC, something which is not what it should be anyways. So this, you have to know, that is 80% of our human DNA variation. And thanks God, it doesn't, all of that has implication on phenotype, because otherwise all of us would be weird. And I have to tell you that concerning the prenatal screening, you have to know that only 10% of the SNPs all along a DNA sequence, they are enough so as to be able to determine the DNA of the origin organ of the origin person. So what was the initial idea of this project is that we have a healthy individual who is in his blood, the little green DNA which is coming from his donor is circulating, and we suspect that in an acute cellular rejection, this DNA circulating will be much more. So then we said, okay, we'll have to apply a test using the SNPs so as to monitor throughout the allograft patient's evolution these levels. So here I used a slide from a heart transplantation paper, which is a really good one, just to tell you how we do this assay. So we take blood, we spin it, we separate the plasma from the red blood cells. Obviously, in the plasma, so in the upper layer, you have the cell-free DNA being of the donor and of the recipient. And on the remaining red blood cells, you only have the genomic DNA, right? Are you all following me? So then so after having extracted the plasma, then we apply all these 41 panel of SNPs, trying to differentiate between what is coming from the donor and what is coming from the recipient. This is quite innovative because until now, there are a couple of studies that I'm happy to discuss with you afterwards of the break in adults that they're using either the whole genomic DNA, which is much more costly and it takes much more time to do it, and some groups, one recently in Spain, they managed to use a panel with SNPs, but they used 240 SNPs, while we are using only 40s. So then we have how we are sure that these 41 SNPs, they are enough to find out which is coming from the donor and which is coming from the recipient. So in order to, let's say, test our specificity and our sensitivity, what we did is that, as I mentioned here, we spike with known concentration. What does this mean, really? So we took, initially, we had to control with genomic DNA. So with known concentrations, we take, let's say, 7 aliquots of me. I am the recipient. I have a kidney from somebody else. We take my blood. We separate in 7 little aliquots. And in each aliquot, we put different known concentrations of the genomic DNA. And then we tested if the results with our model was the same as the concentration we suspected to found, we put initially on the samples, really. Then the other thing we did is that the heart transplants, they have protocol time biopsies. So at that point, when they were doing the biopsy, we were testing directly from the tissue of the donor the DNA. So then we were double checking if with our method we have found the same thing or not. So that was quite useful to use the 3 organs, really. And then we use the statistical model to go from the first step to the second step. So the first step is to be able to differentiate if the cell-free DNA we are finding in our samples is more AA or more AB, so to differentiate between donor and recipient. And the second step is to differentiate to quantify exactly the amount of the donor DNA, which in this case we used in our statistical model, really. That gives us these weird slots for every of each patient. So we have the raw data, then we have the only informative data, and then we split them with a minimal allele, we double them, and anyway, at the end, we have that, which gives us the final percentage of donor-specific cell-free DNA in the plasma of the recipient. So here, with this will make more sense of what I'm saying until now. It's a patient that was transplanted 2-3 weeks ago, a little Krishna. So it's a boy known for valves. He was on end-stage renal disease, obviously on PD, and he had his first transplant from his dad with a very good mismatch, 100. He was under the twist protocol, and here, as you can see in my curve, put the next one, sorry. Yeah, I have put even the creatinine and the EGFR. So that was the levels pre-transplant. The first day, this is the percentage of the donor-derived cell-free DNA in his blood, how it goes up the second day, and how it goes down in the serial sampling that we were doing for the first week after his transplant. So until now, we have recruited almost 140 patients, as you can see from different organs. We have different percentages. This depends, obviously, if you have a deceased or a live donation as well, of how high you will find the detectable levels. And the take-home message is that donor-derived cell-free DNA can be used as something really pioneer marker in solid organ transplantation to monitor the health of the allograft before severe rejection. So that's the purpose of doing it and anticipating rejections episodes and biopsies, doing less biopsies probably. We hope to be able to validate this assay, and the next step, which hopefully will present next year, is to validate with more clinical results. Thank you.
Thank you very much. That was excellent. Any questions? Okay, while people are thinking, how are we going to look at the results according to different organs. So we've got a GFR, so you can have a percentage function. Do we look at the ejection fraction in our hearts? Do we look at the peak expiratory flow rate?
No, but I think that in the initial step that we have to combine all this, I think that we have to sub-specialize for each specialty their own criteria about rejection, their clinical markers, and then just rely on that. But this is, anyway, as I told you, is the second step, because on the first step, I think that we have to increase our numbers in order to improve our specificity of the tests. Because until now, we have 3.7% of undetectable levels, which it might seem to you low, but we have to improve that. So we might need to go up maybe on our SNPs, for example, and use, an idea was to use like 60 instead of 40 in our panel. But we're still going to be pioneering.
Good. Okay, thank you very much for
Thank
that.
you.
That's excellent.